Hey students, good morning everyone. So in yesterday's class, our previous class, we have seen introduction to clinical trials, like why it is important, what it is, what are the factors to be considered when you are doing a clinical trials. Okay, and we have seen this definition. Okay, and in today's class, we will see the types of clinical trials and phases of clinical trials. Right? Okay. So So what are the types of clinical trials or phase of clinical trials? So I think you'll be having an idea about the phase of clinical trials. Like we have phase 1, phase 2, phase 3, phase 4 of clinical trials. And types of clinical trials. Yes, yeah, there are types of clinical trials like intervention trials, prevention trials, or educational trials. So all those things we'll be seeing in today's class, right? Okay. So we have seen all these things in yesterday's class. We have seen the history of it. We have seen when do we have standards. We started preparing a standard documents or following guidelines for clinical trials, and we have seen the basic principles of it. Okay, prophylactic, diagnostic, therapeutic devices, regimens, and procedures. And next, we have seen interventional group, control group, and important factors to be considered in clinical trials, right? And next we have seen all these things, study designs. Okay, so today's class we'll be seeing the types of clinical trials and phases of clinical trials. Okay, types of clinical trials. So what are the types of clinical trials that we already know? We have seen the clinical trial. When you're doing a clinical trial, we should follow one of the any of the basic principles or the considerations, like your treatment should be for a disease condition, or you have a device for disease condition, or you have a new diagnostic approach, or you have a new procedure for any surgical procedure or anything. So like that we have different types of clinical trials, right, that we'll be seeing now. First thing is treatment trials. Okay, in treatment trials, they'll test the experimental treatment or a new combination of drugs or new approaches to surgery or radiation. That means here you are directly preparing a drug, means your clinical trials includes a drug. Okay. Next one is prevention trial. Means here you are preparing a prophylactic treatment, or you are preparing a way to prevent any disease condition. Like we'll be giving, the government will be giving polio vaccine to prevent polio, right? And we'll be giving vaccines to the children to prevent from any of the polio or so many other disease condition, right? So that includes a prevention trials. You are here preventing the thing to be happen. Next, these approaches includes, these prevention trials can be include medicines, vaccines, vitamins, minerals, or any lifestyle changes for any obesity or diabetes or any of those kind of things. Okay, your prevention trials includes, it can be any one of the following thing. Next, diagnostic trials. Okay, so here you are conducting trials to find out a better test or procedure for diagnosing of any particular disease or a condition. Okay, so here you are finding out a better way to see the disease conditions, a better way to find out the disease condition, right? Initial stages of, early stages of disease finding out. So you're developing so many sophisticated methods and techniques to develop it. Okay. Next one, screening trials. So like whenever this screening trials includes people to find any procedures to find out people who are at risk of developing any disease conditions, right? Take example, we'll be saying what you call as people who are having diabetes are risk of developing nephropathy, diabetic neuropathy, means it can affect the kidneys, it can affect the eyes, right? So how they came to know this? They done a screening test or early detection of it, right? Take another example of risk factors. How come we know there is a risk? Means people who smoke, there is a high chance of getting lung problem. How? We have done a study and we came to know people who smokes, there is a chance of lung cancer. People who choose certain good carb, those kind of things, they have a chance of getting any other types of cancer. So here we have done a screening on kind of people who are at risk of developing disease conditions. Okay, it can be those kind of trial. Okay, next, quality of life trial. Okay, here it says about explore a way to improve comfort and quality of life for individuals with chronic illness. Means like whenever you consult a patient, you will be saying about change in the habits. Means it can be about the food habits, it can be about the regular activities habits, so everything you'll be saying it. How come we came to know about it? Because there was a trials, the types of trials happen which will improve the quality of life of patient. Like patient who is obese, he has to walk to reduce the weight. Okay, patient who has a chronic illness conditions, who have a sedentary lifestyle, who have a depressive disease condition, they have to walk, they have to go for exercise. How come we came to know about this? Because the trials are happened on these conditions, trials are happened on these issues, and we came to know this is a better way. This is a change in sedentary lifestyle, a change in the lifestyle modification can improve the quality of life, right? So these kind of things can be happen as a one type of trial. Like we have preventive trials, treatment trials, prophylactic trials, diagnostic trials, screening trials, and quality of life trials. Next thing, this is just a picture from one of the old picture, like how many years it takes and how much money it takes to enter a drug into the market from the discovery phase. If you see this cone shape or the thing, so like I said, whenever we start with the clinical trial, we start with the drug discovery. We start with around 5,000 to 10,000 compounds. Okay, by the time it will reach for preclinical testing, only 250 molecules will reach preclinical testings. Then only 5 molecules will reach the clinical trial phase, clinical settings. Okay, so once this trial will happen and all these 5 drugs, then we'll come up with a new only one molecule as a king, a queen, which will get approval for the from the FDA. At any point of time, at any point of this time, there is there can be attrition, there can be rejection, everything will be gone wrong, and you are going to stop the trials, right? A trial can stop at this phase or at this phase or at this level, at this any level it can be happen. Some drugs got rejected even at the final stage of drug approval also, and some drugs has been rejected even after entering into the market, like your thalidomide which has been developed has been rejected in the market. Where glitazones, some of the glitazones have been rejected in the market because they have completed all these phases, all these levels. They came to get an approval. After getting approval, there are certain side effects. They have to be stopped, right? So, on this graph represents typically an years of time which has to be needed. So, generally, at an average, it will be taking 15 to 12 years or 15 plus years to discover and develop a new drug and release into the market. Okay, and so this is see, this thing won't be appropriate for all the trials. It may vary according to this. They said a drug discovery phase will be having 2 to 10 years, includes your preclinical testing. Then phase 1 will take 2 years, phase 2 3 years, phase 3 3 years, FDA approval almost 1.5 year, and phase 4 4 to 16 the year. But it will take phase 4 is nothing but we are given the drug in the market for use, and we will be seeing side effects of the drug for a period of time, in years of time. So that is your phase 4. Okay, and the cost according to this picture, they said 892 millions. No, it will take almost 3 or 4 billion US dollars to develop a drug. Consider you are to develop a new drug into the market from discovery phase to development phase and usage phase in phase 4, and after that it will be taking almost 15 years of research work of lakhs of people and 3.5 billion US dollars. Consider how much it will be. After spending this much amount of time, money, and lives of so many people, the drugs might reject at any time of clinical trials or after even after giving a shot in the market also. It can be anything. Okay, that's why the clinical trials is being one of the important thing and all. Okay, so we have some other details here, like phase one that we'll be seeing in next slide. So these are the two things which I want to say. This one is a brief about your phases. So phase one will be using 2280 or 2200 healthy volunteers. We'll be finding out the dose to see the safety and PK/PD of the drug. Means to see the pharmacokinetics and pharmacodynamic of the drug, to see the safety of the drug, to find out the dose. So here dose means generally when we see in phase one, what happens? We don't know the exact dose of a drug, right? So the dose may be for toxic effect, the dose may be for therapeutic effect. Means to show any changes in the physiology of the people, we should find certain dose of the drug. So that one will happen here. Phase 2, 100 to 300, safety efficacy will find out. Phase 3, hundreds to thousands, safety and efficacy. Phase 4 is a regulatory approval for post-marketing surveillance, right? So this picture clearly says, if you see this one clearly, here the patients are getting recruited, okay? Then they are being tested for certain disease conditions. Next, disease conditions, and then they're getting results. They're getting certain benefits, which will get approval from the market, right? And the next one, so clinical trials support. So whenever you're doing any clinical trials, you should get support from all of these people, like a monitor, a trainee, let's say about the quality, protocol review meetings. You have to conduct, you have to have a support from contract financial management. You have to have a support from the government agencies. You'd have a data teams, the protocol coordinations, and all these things has to be plays a major role in getting approval from your clinical trial or clinical research support team. Okay, about seeing about a procedure. So this one was about your phase one, ma. So the procedure about your phase one. So this one we'll be seeing in later slides. The slide came earlier. Okay. So phase of clinical trials, as you know, we have 4 phases of clinical trials. Okay. So everything should be approved from the national regulatory authorities like DCGI and CDSU in India, ICH, GCP or FDA in US, Europe, Australia, right? Yeah, this is same again. The 4 phases of clinical trials. Like in each phase, we'll be seeing phase 1 will be seeing safety parameters. Phase 2 will be seeing effectiveness parameters. Phase 3 will be seeing confirmatory. Phase 4 will be seeing, like it's obviously the drug in the market, and we'll be seeing the effects drug in the market. So one of the new point which I have to show in this slide is approximately 70% of the drugs from phase 1 will move to the phase 2. Remaining 30% of the drugs will be rejected, which will be kept in the compound library for some other use. In phase 2, only 33% of drugs will pass from phase 2, which move to the phase 3. See the difference. Out of 100, only 70% of drugs will clear phase 1, and out of it, only 33% of drugs will move to the next phase from phase 2. And in phase 3, only 25 to 30% of the drugs move to the next phase. Okay, so this was the article which I've taken from US FDA. It was one of the obviously not the recent article. It was way back in 2000 between. Okay, next one is phase 1, right? So this picture says a lot, like where we are taking informed consent for the patient. We are doing examinations with his blood levels. We are inducing drug to him. We are collecting the samples. Everything will be done by a clinical investigator. Okay, right. So yeah, phase 1. So phase 1 is where we'll be seeing a group of people of 20 to 80. Means here we'll be taking 20 to 80 healthy volunteers in phase 1 trials. Okay. So what is exactly healthy volunteer in clinical trials? We'll be seeing in upcoming slides. Generally, according to us, healthy volunteer means who don't have any disease condition, who are not obese, whose BMI is appropriate, whose weight, height, and everything is appropriate. We call them as healthy volunteers. But in clinical trials, we have a different definition for it that we'll be seeing in upcoming slides. Okay, next one. So phase one will be done to see safety dose, tolerability dose, pharmacokinetics and pharmacodynamics of the drug. So this PKPD, tolerability and safety, we will be getting in preclinical trials, but preclinical trials will be done in animals. So we'll be getting the safety, tolerability and PKPD in animals. We don't get in humans. So to get those things in humans, we'll do phase one. So what we'll do is we'll extrapolate the dose from animal to human, right? Like supposing if 1 gram is a safety dose, a tolerable dose in animal. So what we'll do, we'll start with 2 grams in humans. We'll do the dose extra is a dose calculation we'll be doing. We'll extrapolate the dose and then we'll use in humans to find out these parameters. Okay. Next, volunteers. So volunteers, like we say, healthy volunteers means who don't have any specific disease conditions. Okay, so they are paid for their inconvenience fees. Okay, for their time spent in volunteer center means if I'm asking you certain help, you should get something back in return. Then only you can help me, right? It's not just for silly things, but speaking to the clinical trials. So volunteers are those people who be a part of the study. So whenever being a part of the study, they should get certain benefits, right? What are those kind of benefits? Okay, so that we'll discuss in detail in upcoming slides. Yeah, like I said, only 70% of the drugs will pass through this phase one. Again, in phase two, only 33% means now 70% will become 100% for phase two. Among that, only 32 will go for phase two. Among those 33, the 33 will become 100 for phase three. Out of that, only 25 will reach your phase three, right? So, sorry, phase four. So, like this, there will be attrition rate. So phase one, so what is the procedure for phase one we'll be seeing now? Yeah. So procedure for phase one is subject recruitment. You'll recruit the subject. Next, you'll take informed consent form from them. Okay. Next, you will screen the patient based upon the health condition. Next, you will record the baseline parameters of the patient. Okay. Next, you will administer the drug to the patient. Okay. Next, you collect the blood samples. You'll administer the drug. You'll collect the blood samples to the patient from the patient. You'll record the post-treatment parameters of the patient again. You'll record it. What are the changes happened in a patient? You'll record it. And next, you will analyze the blood samples for the given drug levels. Like this is like a Cmax, Tmax, and the dosage levels which will be seen. Okay, and then you will analyze the data whichever you have collected. You will analyze it after giving drug. What kind of changes you have seen in a patient? What are the systems, like different systems, like your different organs in the body, which have been affected by the drug, like your PKPD. So that will be checked in this procedure. Okay, so we'll discuss in detail about this after some time. So now going back to your objectives of phase one. The first objective is dose safety, right? To determine the most significant adverse events in human subjects, right? Like we said, the safe dose of paracetamol in humans or the safe dose of ceftriaxone in human per day is 2 grams. More than that is a little bit toxic, not much. Like, and the safe dose of paracetamol in humans is 4 grams per day, who don't have any liver problem. So that is a safe dose. So those kind of safe doses will be checked in phase one. Next one, tolerable dose. So tolerable dose means the safe dose range is determined by the dose calculation and corresponding serial lab test. So a tolerable dose means, supposing the tolerable dose of paracetamol in humans is 4 grams per day. More than 4 grams of paracetamol in humans is a toxic dose. Means till 4 grams a human body can tolerate the effect of paracetamol, right? That is the tolerability dose. So this one also will be seeing. See here we don't see the therapeutic effective dose. Instead we'll be seeing the maximum tolerable dose. So how much dose a drug a body can be withstand. Like take example of your X-rays. So when we say X-rays, there is a wavelength which will pass through our body, our system, right? Right? How can we came to know what wavelength should be passed through the system or our body? So through X-ray, through the series of experiments and clinical trials, wherein which a different wavelengths will be used on different people in clinical trials, and then we have a setup of range, wherein which okay, this wavelength to this wavelength, the rays can penetrate thoroughly through the human system without any issues. Like we cannot say without any issues. Even there are negative side effects for X-rays. I don't know whether you know it or not. Like once you undergone your X-ray, how much effect you'll get with that X-ray means when there is certain radiation passed to your body, definitely a minute level of toxic effects or unwanted effects will be there. Okay, so once you gone through X-ray, how much effect you'll get to your body is equal to a layman who work in a sunny day for entire day. Like when you work hard in a sunny day for entire day, how much toxic effect you'll get. You'll be saying, okay, how much what toxic effect we'll get when we work under sun. It's good for health. That's good for health, but you'll be getting certain one or a minor level of toxic effects, right? So it means how much radiation will penetrate into your body when you work under sun for an entire sunny day. So that much radiation will be getting into your body once you undergo your X-ray. So next time, make sure when you're going undergoing X-ray, make sure is it necessary for you or not. Okay. Next is PK and PD. What drug is doing to the body and what body is doing to the drug, right? What body is doing to the drug and what drug is doing to the body. Pharmacokinetics and pharmacodynamics we'll be seeing in this phase. Next, prerequisites for our phase one means. So what are the things? Suppose consider you want to take admission into any of the course in any college. Your prerequisite is your previous year's percent. Like if you want to join a degree, you should have certain percentage in your plus two. If you want to join into your masters, you should have certain minimum percentage of marks in your degree. So like that, what are the prerequisites for any of your connecting clinical trials in phase one means one of the prerequisites is preclinical safety data means. So preclinical safety means we will be getting certain safety data from your animal studies, like your preclinical studies, right? So we'll be seeing the pharmacological approach and toxicological approach. Okay, we'll be seeing data from their single dose toxicity studies, repeated dose toxicity studies, pharmacological studies, local tolerance studies, pharmacokinetic studies, mutagenicity studies, and repeated toxicity studies. All the things which were the data available from the preclinical phase, we'll be collecting it. Then we'll go for the phase one of clinical trials, right? And next thing is regulatory authorities and ethical committee approvals, right? So this is the data which we need to do the study. And next, that data should be submitted to the ethical committees and national regulatory bodies to get approval for conducting or to start a clinical trials. Okay. Next one is conduct of the clinical trials. So where are you going to conduct clinical trials? So obviously you have to conduct it in a trial site or in hospital or in a certain laboratory units, right? So those things are called as clinical pharmacology units or clinical trial units, right? Next, where the subjects are being observed for the by the full-time staff means here the people will be there under the supervision of doctors and nurses and pharmacists or some other caretakers, and the entire supervision will be there means the patients will be there in the trial site for the entire time of the drug being used for that patient. Okay, so what are the equipment should be had by that laboratories where the clinical trials are happening? Okay, all types of physiological functions and drug levels in biological functioning means the trial site should have capacity and facilities to do all the physiological functions and to find out the drug blood level means like Cmax and Tmax or the drug levels in the serum and blood that should be found out by the clinical trial site. Next one, you should have facilities to handle the emergencies. Like Being phase one of clinical trials is being really a tough time because we don't be having much data here. We'll be having only limited data. The two from animal studies, we don't have 100% data from human studies also. So we should be we there might be a chance where we'll exhibit certain toxic levels or areas to the patient. So how do we handle it? How do we handle those areas? Supposing if patient has developed anaphylactic or anaphylactic reactions, or patient has developed certain life-threatening conditions because of the drug, how to overcome that one? We should have a standard protocol for it. Okay, and the trial site should be having a emergency unit, all the facilities to avoid and to take care of this handling emergency condition and unexpected adverse drug reactions. So we have seen a statement: we should not expose people to unnecessary risk. That's right, but we don't know all the data at this time. So we are going to obviously expose people to certain risk. Okay, but that should not be unnecessary. Means we know it and we are doing it. That is we are in that time, we know and we are doing it. We are exposing people to unnecessary risk. That is an issue. And if we don't know and we are doing it, that's not in our hands. But if anything happens like that, we should have a team to take care of them. Like now we know anti-cancer drugs have so many side effects, but previously we don't know it. At that time there will be phase one, phase two trials also for chemotherapy, for cancer drugs, anti-cancer drugs. But now we have a phase one for cancer, anti-cancer drugs, but we don't take healthy volunteers. We'll be taking patients directly, or we'll stop doing phase one. We'll go for phase two directly. Okay, like the laboratory should have a facility to process the blood samples and plasma samples and all to see the drug levels, PKPD. Next, who are all the players which plays a major role in clinical trials? Means the investigator. Okay, so the person who usually conduct the clinical trials, we call them as clinical pharmacologist. So here the clinical pharmacologist is nothing but a doctor. Okay, apart from that we need medical staff, paramedical staff should be qualified and skilled enough to handle any kind of emergencies. Suppose if there was a hypersensitive reactions, like the patient has stopped the breathing, or there was a rashes appear all over the body, so you should have sufficient people to take care of those basic life support and advanced life support. Next one is the subjects. So at this phase, the subjects are nothing but your healthy volunteers. Okay, so and sometimes you can include a patients in cancer trials. Means like being known we have severe side effects for this class of drugs, and if you want to include healthy volunteers, it's not a good thing. So our the clinical trials guidelines and all, they will stop. They recommend, they say we are not going to, you cannot include healthy volunteers in phase of clinical trial, first phase of clinical trials for anti-cancer drugs. Next, so what is a healthy volunteer, healthy subject? According to WHO, a person who is free from any abnormalities that would complicate interpretation of the data or increase the sensitivity of the subject to toxic potentials of the drug. Means here they are saying, supposing you are doing a trial on your arthritis. Consider you are doing trials, your drug is an anti-arthritic drug, or you are finding out any NSAID drug. Okay, so if you need a healthy volunteer for it, it's okay. If you take a person who is having your hypertension, you can take it, because we don't have much link to hypertension and joint pains or for arthritis, right? So that can be, that person we can call as a healthy volunteer, because here the person don't have any problem to the bones or to the joints. You can take that person as a healthy volunteer. But if you take a person who had an accident 1 year back or 2 years back, we cannot consider that person as a healthy volunteer, because his joint pain or his joint problems can lead to a problem to you, can give a wrong results to you, right? That person is a healthy volunteer, because if you take that person, there will be an increase the chance of sensitivity to that patient. Okay, and next one is the summary of main points raised in favor of healthy volunteers as scientific benefits. Means if we take healthy volunteers, we don't have much work to be done, that much detail. Next are potential benefits, because in healthy volunteers we already know what are their his problems, and we already know the physiology of the patient, so there won't be much things to be taken care. And regulatory benefits, even the regulatory authorities don't ask for much data whenever he's going and using regulatory benefit kind of things and all. So that's what it is. Yes, ma'am. And next thing here is that, yeah,